My name is Lyon. I'm the laboratory director at KCAS Bio, Lyon, France. And I'm going to take you a tour of our facility. We are on the third floor, which covers 1,000 square meters, and this is the office. And I will show you our laboratory. We are close to the laboratory. Samples arrive through the delivery entrance and they're handled by our logistics team. Access to the laboratory is secured by a badge and only for authorized personnel. In this room, all samples or shipments arrived, they are all recorded and checked to ensure the sample integrity during the transport. Then, samples are unpacked and stored in an appropriate location in the storage area. This main corridor provides access to all of storage area for sample and reagents. Here it's our room temperature storage area where we store every plastic and reagent for the assay. We have a large cold chamber to 2 to 8 degrees when we store all of the reagents. Behind this door, we have a central storage location for sample with 780- freezer, 4 liquid nitrogen tanks for a total capacity of 200,000 samples. Each location is monitored by your temperature control and alarm system. The building has an emergency power supply backup with a generator and a UPS system. Once samples have been received, they are recorded in our Watson-LIMS system and they are released for the analysis. And then can go to our BCL2 laboratories. Our laboratory is divided in 3 labs dedicated by assay. One for the ligand-binding assay as AB, LBA with several immunoassay instruments. Another is dedicated for cells-based assay as ELISpot assay or cell culture. And the third one is dedicated for flow cytometry. Each lab has their own pre-analytical and sample processing area. Here we are where we prepare sample with a liquid sample handling system that we have here, or for tissue extraction. It's also the lab where we produce our specific coupling reagents for the method that we develop. Here is the lab where we have all of the instruments for the LBA assay. Depending of the marker of interest or the level of sensibility, we can offer a large variety of instruments in this lab. We have the new LISA technology with an Argo instrument, 2 Luminex platforms that we can run in the same time up to 20 markers. We have a classical microreader instrument, 2 LR system when we can run multiplex assay with a semi-automatic system. The MSD, we have 2 MSD, we can run high-sensitive assay or multiplex. Behind this door, we have 2 automatic systems for immunoassay instruments as the LumiPulse for IVD assay and CMO HDX for high-sensitive assay. And I want also to show you this one. It's the Immunospot reader for cells-based assay as ELISpot, and made the transition to show you our second laboratory for cells-based assay. So here is the lab dedicated for cells-based assay. As you can see, I hire Protect to ensure our sterile condition required for the assay. We have a typical equipment as a biosafety cabinet, a centrifuge. We have a CO2 incubator for cells grow, and a cell culture. In this room, we prepare our PBMC for the assay, but also we process all of the immunospot assay. Here, the lab that I want to show you is dedicated for flow cytometry. In this room, we process our blood fresh sample by a dedicated analyst team. They are processed and stained on this lab and after acquire of several instruments that we have in this lab. We have 2 BD for TESA for conventional flow cytometry and also 2 Cytec for spectral flow cytometry. All the instruments are standardized across the site with Philadelphia and Melbourne to ensure for our sponsor that we harmonize the analysis and the results. As you've seen during this tour, our lab have a high capacity for a ligand binding assay with several platforms and for flow cytometry. All analysts are trained and qualified to use the instrument. Each instrument have the redundancy and backup, and our site is GLP/GCP compliant. I hope that you enjoy your visit at KCAS Bio Lyon, France, and get the chance to visit our location or other location based in Kansas City or Philadelphia.